We're live.
All right. So for the first question with a disease called Angelman syndromes, for A, we had gotten the answer right here. Right here. They're homozygous dominant. Well, sorry. What happens is the phenotype for all of them they would all have Angelman syndrome. And the way we got to that and the genotypes is you would do a cross. And with me, I labeled which allele came from which parent. So in this one, the male got the dominant gene and the female gave the recessive gene. And then since with the Angelman syndrome, the paternal inherited alleles are shut off, I crossed out all the alleles that came from the father that were shut off. And so all of them technically would be recessive. And so all of them would have the disease. I started off with B. So that was B. A, so half has the Angelman syndrome and half has the normal phenotype. And I did the same thing, figured out what was male and what was female, and then crossed out all the male ones, and half of them ended up with the dominant allele and half of them with the recessive allele. And so that's for A. B what was first. With C, with the triplo X syndrome, we got that there were 0 Barr bodies formed because at least two XICS genes must be present for Barr bodies to be formed. And because of that, there was only one in there. And with that one, it wasn't enough to give any Barr bodies. So we got 0. For D, we had gotten 1. Yeah. 1 Barr body because T6 and XIST work together to create a double-stranded RNA, which prevents the formation of a Barr body. And so with the 2 deletions there, it prevented the Barr body from being formed, and we ended up with 1. For number 2, we got SRY is an example of a holoandric gene. MYC2 is an example of a gene found on both X and Y chromosomes. For C, the answer we got was half dwarf, half normal. For D, in sex-influenced pattern of inheritance, an allele can be dominant in one sex while the same alleles is recessive in the other. Genomic imprinting is an epigenetic process that occurs during gamete formation, while dosage compensation is an epigenetic process that occurs during embryogenesis. For F, the answer we got was XIST and T6. They, just like in 1D, they work together to form a double-stranded RNA. G, we got for the first autosomes, and the last is X chromosomes.
Okay, my turn?
Yep.
Yeah.
Sweet. Okay, so this problem's talking about this terrible deficiency that relates to mental deterioration if not properly treated. So it's talking about how this woman has this OTD deficiency, and that it's an X-linked recessive disorder. So and it also gives us the information that the woman's mother is homozygous and unaffected. So that means here, so for A, what is the genotype of the woman's father? So if the woman's father is affected by this deficiency would have, being a male, he only has one X chromosome, therefore he would have to be recessive for that in order to be affected. So his genotype is X recessive Y. And the second part of that question is, what's the genotype of the woman's mother? And we see that she is homozygous and unaffected. So she's got two X chromosomes and both are dominant. Okay, so for B, the question is asking if the woman has children with a man who does not have OTD, what is the chance that their first child will be a son with OTD? So we have to take into the account that when you do a cross between a male and a female, that their offspring, their progeny, are going to be 50% female, 50% male. So you have to start out with that one first. So you get 1/2 chance of being a son. And then you look and do the cross. I did my cross up here. So I crossed the male who was unaffected. So he's got the X dominant gene Y. Y doesn't carry the gene. And then with the woman who has and her genotype was X dominant X, so she was heterozygous in overall. So the chance of them having a son that's affected, so you have the 1/2 chance of being a son, multiplied the 1/2 by the 1/2 chance of being affected. So there it is. There's his genotype. So he's got that 1/2 of males being affected. So I got 25%. So there's a 25% chance that their first child will be a son with the deficiency. So C is talking about it wants us to identify the genotype of a male with whom the woman could produce a daughter with OTD. Okay, so we gotta do another cross here. And the daughter, in order for a daughter to be affected by the deficiency, she has to be homozygous recessive, just like right here. Shows it right here. So you have to do the cross between a male that is affected with the recessive gene with a. So I guess. So I guess you could either have a woman that was affected as well, or a woman that was heterozygous. Would get you similar results. In this case, I crossed a heterozygous female with an affected male. In that case, for D, you would have to I wrote it out this way. So 1/2 of females affected would be homozygous recessive, multiplied by 1/2, or 1/2 of the females would be heterozygous. So in order for that to happen, you would have, so the genotypes and phenotypes of the offspring, so you would have 1/2 of the females would be affected, whereas the other 1/2 would be heterozygous, and 1/2 of the males would be dominant and unaffected, whereas the other 1/2 would have the recessive gene and be affected.
Okay, so for question 4, it says the presence of a beard in some species goes to determined by an autosomal gene that is dominant in males and recessive in females. So for part A, the genotypes for the male and female for both parents are heterozygous, and the phenotypes is the dads have the beards and the mom don't have the beards. And the way we figured that out was by doing a cross. So it'd be here. And so you just make both parents heterozygous, and then you cross them, and that's how you get the genotype and phenotype. For part B, is asking for a 1-to-3 ratio. And again, doing that, the Punnett square, we also got the same answer, so they're both heterozygous. For both parents are heterozygous for C, a supposed cross between a male with a beard and a female without a beard produces one beard and one no beard phenotype. And for that, we got the father being a heterozygous dominant, and homozygous dominant, I'm sorry, and the mother being heterozygous. And we're just figuring these out by doing the Punnett squares. And also because it says that the presence of the beard is dominant in males and recessive in females. And then for D, suppose a male and female both which lack beards produces a one beard, one no beard ratio among the male offspring. And then the genotypes that we got that once we crossed them together was a homozygous recessive for male and a heterozygous for female.
All right. So question 5 involves X-linked inheritance and reciprocal crosses. So in cats, the orange coat color is determined by the recessive B allele, and the black coat is determined by the dominant B allele. The heterozygous condition produces the phenotype known as tortoiseshell. Both alleles are X-linked, so we crossed an orange male cat to a tortoiseshell female cat. The genotypes and phenotypes that we get, so we get one heterozygous female, so she would be tortoiseshell. One recessive female, she would be orange. One dominant male, he would be black, and one recessive male, and he would be orange. Question B concerns the trait in part A. So male tortoiseshell cats are very rare. Their genotype would be, they would have an extra X chromosome, so they would have both the big B and small b on separate X chromosomes and the Y chromosome. And that is why they are very rare. So in the fruit fly, a mutation produces the scalloped phenotype. So the scalloped phenotype is a result of an X-linked recessive mutation. The genotypes and phenotypes of the progeny when reciprocal crosses are done is the females would both be heterozygous and the males would both be they would not have the trait. And so then the phenotypes, they're all unaffected. So D is, this question concerns the scallop phenotype described in part C. The scallop phenotype is the result of an autosomal recessive mutation. Indicate the genotypes and phenotypes of the progeny produced when reciprocal crosses are done. And so how you do it this way is you take either a homozygous recessive mother and then a homozygous dominant father and do the crosses, and you would get heterozygous for all the children, and then you do the same flipped, so the mother would be homozygous dominant and the father would be homozygous recessive, and you get the same results. So all 4 of the progeny would be unaffected of scalloped wings, so none of them would have it. All right, and that concludes our video.